Welcome to the Healthcare Technology Report, a leading information source on healthcare technology companies and solutions. In this episode, we speak with Ola Wlodek, CEO of Constructive Bio, a company developing technologies for large-scale genome writing and genetic code reprogramming. Spun out of Professor Jason Chin's laboratory at the MRC Laboratory of Molecular Biology, Constructive Bio focuses on building programmable biomolecules and enabling cells to produce materials and therapeutics that conventional biology cannot. Ola brings experience from across the Cambridge biotech ecosystem and previously served as Chief Operating Officer at Reflection Therapeutics. At Constructive Bio, she leads efforts to advance applications of synthetic genomics, from virus-resistant production strains to the biosynthesis of new classes of enzymes, pharmaceuticals, and biomaterials. I'm your host, RJ Lumba. We hope you enjoy the show. If you like the episode, click to follow. Ola, thank you so much for taking the time. It's a delight to be with you.
I'm very excited to talk today. Thank you.
This is a fascinating area that you're working in with DNA sequencing. And what you do in particular is very specific. It seems daunting to the average person that you could actually, in effect, change the genome in the way you do it. Maybe tell us a little bit about the technology.
Absolutely. I think we've all heard many times about the molecular biology, so the technologies that gave rise to companies like Genentech and us being able to create programmable molecule where we use DNA to make products that change patients' lives. But what we're doing is a molecular biology 2.0. So what we decided to do is to write entire genomes from scratch, where we take entire control of the genetic code. And that allows us to change the palettes of the monomers or the building blocks that proteins and peptides are made of. What it gives us is an ability to decide and design what those molecules will be made of. And as a result, we can manufacture molecules with new to nature features, but exactly in the way that biology intended it to do. So we can encode in DNA and produce in those green cell lab biofactories that we like to call the cells as any polymer with full sequence control. This is a fantastic technology which came from the academic lab of our founder, Jason Chin. But what we do at the moment as Constructive Bio is we program peptides and proteins with non-canonical building blocks, and we manufacture results of that programming at scale.
And where is this healthcare technology currently, or biological technology currently used?
So I'm going to talk about the outputs of such technology. I think we're living in the era of peptide drugs. Everyone at the moment is excited about Ozempic and Mounjaro and a number of other clinical candidates. And I'll come back to this in a moment. Mm-hmm. But those drugs have been transformed by new to nature building blocks. They have this small monomer called 2-aminoisobutyrate, and that means that the patient's bloodstream doesn't chew through those peptides, okay? And as a result, the patients can have an injection once a week, and their appetite control and their metabolic improvement happens week after week of the treatment. So there are on the market some very amazing drugs with non-canonical building blocks. And in fact, there is more than 100 peptides with non-canonical building blocks already approved and on the market. But there is a caveat. Until today, most of those peptides have been made through solid phase synthesis technology, which is essentially chemical manufacturing. And while it is okay to make a small smidge of peptide this way, when you start producing at scale, for every kilogram of synthetic peptide, you produce 13,000 kilograms of chlorinated waste. And it's a pretty horrible prospect, because I don't know about you, but I would not want to take something that maybe will be good for me, but is going to completely destroy our environment outside. And I think there's lots of people who, if they want to subscribe for a long-term need of a peptide drug, would like to ask themselves, how is this drug made? Okay. And that's a kind of side effect of where biology is so much better than chemical synthesis. And I think you can ask every child at school about chemical synthesis, and the initial association is fuming, dirty, horrible factories. While in fact, biomanufacturing gives us a green, scalable, cheap manufacturing, because that's what the life is all about multiplying and filling the big vat with bacteria. So what we're doing here and what the real unlock is our genome recoding or synthetic genomics that I've talked about earlier allows us to close the gap between designing really amazing molecules, programming them through DNA, and then manufacturing them at scale through the green, fantastic bioprocess.
Mm-hmm. I'm curious if there's any loss in efficacy or downsides to biomanufacturing compared to what's currently being done.
The biggest loss is that you have to commit to a new process, right? We know how chemical synthesis works. You create a reactor, you bring in all your peptide blocks in it, you create the peptide, you purified it, and you're good to go. So people have been good at doing that since 60s. While biomanufacturing has all these advantages, but it's not exactly a trodden path. And I've mentioned Genentech in the beginning of our conversation.
Mm-hmm.
And, they have been producing insulin and other drugs and then antibodies now for decades. So people do use biomanufacturing, but no one has ever bridged that gap between biology and chemistry in a way that our founder did, Jason Chin. So I think the biggest downside is that you have to commit to an entirely new process.
Mm-hmm.
If you do, you will get better cost of goods, better scalability, better greener credential for those who care about that. And I think this is really fascinating.
Can you tell us a little bit about where the company sits today in terms of its life cycle? It seems like you have a team of folks and perhaps you're still earlier in terms of commercialization, but I could have that wrong.
No, you're absolutely right. We are a Series A company. We've raised a Series A in September last year, so slightly more than a year ago. And as a startup, we are very early in our journey. So as of today, we have a team of 50 folks in Cambridge, UK. We have a nice facility here that we are very proud of. It has got some bioreactors, it has some labs, it has some desks. And, we are still in the process of proving ourselves as a company, not in technical terms, because our technology has been developed in our founder's lab a long time ago. We have proven that it works. We have scaled it up and now it's flying. But as a company, we are very open to think about our future. We want to have right partners who will scale that technology with us. And we don't yet have any products on the market. So as a Series A company, we are drafting our future path, but all the commercial introduction is in front of us. We have had some collaboration with a range of pharmaceutical partners around a range of products. Those steps have been going pretty well, and we're very excited about how this is going to grow with us. But we are not yet a large-scale manufacturer, although we do believe the technology is ready and we want to be.
You mentioned partnerships, so presumably the drugs would be developed by the pharmaceutical company, but using your technology. Is that correct?
Yes, that is correct. So we want to enable the partners with our amazing tech and those processes that I've mentioned. But we are a drug discovery company in a sense of the molecule, but we want to partner with people who understand deeply the cellular biology and the disease-related needs. So at the moment, partnering is super important for us because we want to equip the partner with that amazing tech. We can discover peptides and proteins with new-to-nature features. We can make them at scale. We can license out that fantastic manufacturing protocol. But the partnering is what's going to amplify our potential for success.
Mm-hmm. Now, tell us about your background. I think it would be very difficult for someone to lead this effort without having the type of background you have educationally and professionally. So tell us about yourself.
I wouldn't probably be as excited about this technology if I didn't have that background. I have a PhD in biological sciences. I have worked in the labs for most of my, professional life. I've been a very happy scientist, but at some point I've started seeing that I'm perhaps more effective as a leader facilitating the work of others rather than just a scientist with a pipette in my hand. Mm-hmm. And I've at that point decided to strengthen my background with business education as well. But yes, I'm a scientist. I'm super excited about biology. I love synthetic biology. I've worked in making peptides using engineered biological systems for many, many years. And I think this technical understanding of what we're doing is my superpower. Not only mine, but also my other colleagues. Like my company is pretty heavily populated by people with beautiful brains and a lot of scientific expertise. And I think this matters in biotech. I perhaps have opinion that is currently not very popular, which is that the tech is the core of what matters. Because yes, you can waste an opportunity of an amazing technology with poor business decisions, but business decisions on their own are never going to be able to provide people with a revolution, that roots itself in biology or chemistry. So I think my tech background is very important for the company, but I also have a lot of commercial hunger for scaling up this business. So not only scaling up bioprocesses, but also scaling up the company.
Are there competitors? Are there those that have a similar technology?
There are people who have some aspects of overlap with us. And I think, if we didn't have competitors from technology perspective, that probably would mean that we're doing something that is not of value, right? So there's a number of cool competitors who use cell-free processes, and they are interesting. They provide you a broad variety of new to nature features in peptide formats, but they do not bring to the table ability to produce them at scale. So discovery, yes, manufacturing, no. When chemistry becomes programmable like biology, and biology handles the scale, this is where you get the pipeline moving fast from discovery to manufacturing. So our competitors have got great breadth of molecules, but they don't have that manufacturing advantage. And in biotech, everyone competes on the product. So we need to make sure that the use of new to nature features is something truly enabling and not just a molecular biology party trick. So fortunately, we have enough competitors to compare and contrast ourselves and to think about our differentiation. But I'm very proud of our tech and our approach to scalability.
Which near-term milestones are you most focused on?
So we had a very successful technical few years, and now it's all about commercial introduction. So I want to finalize this right deal with a pharma partner. We have a few of those conversations going, and proving ourselves as a business is what really matters at the moment. We've generated some revenue in the past year. We have started those collaborations going, so it's not now about the next step. And that's super important. However, there's also a technical, very important milestone ahead of us, which we hope to hit by the end of this year. We are writing what we are calling a Syn61 2.0, which is a strain with recoding, but with some additional superpower of robustness when it comes to scalability. So that is something very important to us at the moment. We also are running a number of confidential but very ambitious programs around new to nature chemistries. So tech is going well. Now it's about commercial introduction.
We alluded to kind of capital, and you referenced the Series A stage that you're at. How much capital do you foresee needing? Typically biotech does require a lot of capital. So curious as to the next phase.
Of course. So to date, we have raised $75 million, which is a lot, especially for the company in UK. And that gives us a comfortable runway that will allow us to deliver the technical milestones ahead of us, as well as we hope the commercial milestones. And then we're going to see what the future brings. Raising capital has not been that easy in the recent years, so we are in a very comfortable situation as a company, but we will need further funds to grow. My ambition is to perhaps partner and bring revenue from commercial products into the company rather than rely solely on the venture capital. But as you said, biotech is costly and we need to have a discipline about spending and commercial discipline to grow.
We're entering the tail end of the conversation. I have two final questions. One is, can you tell us about a person who has had a profound influence on you?
I think the person who changed me most was my first manager in biotech. So I came to biotech from academia, and I have to admit I was a little bit lost. You grow all your life thinking that you're going to be a professor, and then you finish your PhD and you ask yourself what I'm going to do with myself. And I kind of started my biotech journal through perhaps a bit of an accident. And I met the really inspiring woman, Dr. Anna Stanley Smith, who has injected a love of biotech into me. And I think she was one of the most transformational people on my track. I don't want to give you cliches like being inspired by Jason, our scientific founder, in his intentionality and everything he does on a day-to-day basis, or some amazing colleagues from my work. They know who they are. They're fantastic. But I will give a shout out to Anna on today's talk. But she injected me with a love of biotech.
Last question. Can you tell us what you like to do outside of work?
Oh, I like to lift weights. Gym is very important to me. I think I will go pretty crazy if I didn't have my gym time in right amounts. And I also love walking my dog and reading.
Wonderful. Well, Ola, I want to thank you again for taking the time. It's been a great conversation.
Thank you very much. It was lovely to talk to you.